Hey man, how are you doing?
Tired. It's been a long week.
Yeah. Yeah, I'll be right back.
Yeah.
Okay. All
Yeah, I
right.
haven't done a lot of studying this week. It's just been crazy with work. But I
Yeah, of
did
course.
yesterday I did just a section of Altius for biochem, and I used
Yeah.
the strategy from the video.
Yeah.
Um did pretty well. I got a 125 on it, and I did
Nice.
miss some things. I was after working all day too, but it worked. I had about 9 minutes left after I was done to go back through the flagged questions. And I did change at least three or four to correct after doing that, so that definitely helps.
Nice.
I made a couple of just dumb mistakes after. I kind of reviewed it a little bit. We can go over some of the harder ones. There was
Sure.
one passage I don't feel like immunology comes up that much on the MCAT, but there was a couple two really difficult immunology passages.
Yeah.
Yeah, but I don't know. I just know I took FL2 last weekend, and I was a little bit tired, but I didn't do great. I think I got a 495, and I haven't
Yeah.
even
Yeah.
reviewed it yet because I feel like it was kind of a half-assed attempt.
Mhm.
I think I was just tired and I was running out of time
Yeah.
across
Yeah.
the. So maybe I'll try it again this weekend since I haven't reviewed it and just use the timing stuff. Um, yeah, because I did really bad. And I feel like there's something wrong with my approach because my scores just haven't really gone up much in a year, even though I have mastered a lot of content.
Yeah.
So it's kind of frustrating.
Yeah. Yeah. But yeah, remember, it's not like they don't necessarily just test you on content, right?
Yeah,
But yeah, hopefully that video was useful because I was doing the full length too, and I basically was if you saw how I answered those questions, if anything basically took longer than 30 seconds, I just guessed, flagged, and moved on.
yeah, and I have a bad habit about that because the thing is, I usually can, not every question, but a lot of questions I can get it right, but sometimes it takes me too long to
Yeah,
first
yeah,
go through. It's I'll waste 4 minutes on a question,
yeah, yeah,
and I have three passages left that I'm rushing through and getting a lot of crap wrong. I
yeah.
actually noticed on cars, I haven't reviewed it, but I got the first 10 questions, I missed one of them.
Mm-hmm.
And I started to fall apart halfway through the test because my timing got off. So the nerves kicked in, and then
Yeah,
I was rushing, not comprehending. So then I noticed my accuracy dropped
yeah,
heavily.
yeah,
So
yeah,
the last three passages, I was just guessing on all of them.
yeah.
So that was good.
If you basically you're going to get to the point where if you don't want to spend 4 minutes on any one question because every question's worth the same amount of points. If anything takes long, just guess, flag, move on. Get as many questions complete as possible before you attack those questions again, because that's going to affect your psychology, because you're not going to be as your anxiety isn't going to be as bad because you're going to come back to those same questions that gave you some trouble at first with the confidence that you've already answered a ton of questions.
Yeah. The only issue I ran into with that strategy the first time I did it, I probably still stayed on some questions too long.
Yeah.
There was a couple that got me and I actually got them right, but it did take me a little longer. So I noticed I had about 9-ish minutes coming back to it. But what happened was I got through the first, I think I flagged 30-ish questions.
Yeah,
And when I
that's
came,
okay. I flagged a really high number of questions as well.
so the problem was when I came back to them, I couldn't get to all 30 flagged questions. Like there was a couple I came back to like the first two. I was like, okay, I'm pretty sure that was answered. Let's not waste time on this question. I think there's some other ones I definitely got wrong.
Yeah.
So I would like skip those and just kind of quick check. But there was a couple like towards the middle. And I think it's where I started to get worried about time and my comprehension just wasn't as great.
Yeah.
So should I try to save a little more than 9 to 10 minutes? Like I don't know if I can get them any faster. If I have like 30 flagged, like
So,
I got through like 15 in 9 minutes. Like
yeah.
Maybe I could try to save a little more time elsewhere, but I also don't want to.
So when you review the flagged questions, right? You're doing like imagine doing like multiple passes. So like when you first do the first pass, going through the flagged questions. Let's say you have 30 flagged questions. You're going to answer. You're going to do the same thing. If a question is taking too long, you're going to keep it flagged and you're going to move on to the next one. And so in that first pass that you do, that first review pass, you basically are answering like the easier questions again. So basically, like imagine this, like the first time you start a section, you're going to flag the ones that take longer than 30 seconds, right? And then when you review the ones that you flagged, if it takes longer than a minute, you're going to keep it flagged and you're going to answer the ones that take less than a minute. So that's going to be your first pass. So that 30 flagged questions will go down to like maybe like 20. And then you'll do a second pass where you'll answer questions that'll take like a minute. If it takes longer than a minute, you keep it flagged. And then in the third pass, you'll do the ones that take longer than a minute. This way you'll be, it's kind of like Anki in the sense that Anki shows you the ones that you have trouble with more than the ones that you don't have trouble with. It's kind of like that. You're doing the ones that you find easiest first, then second easiest second, third easiest third. So you're basically just doing like these passes.
So go ahead, sorry.
Oh, and you know, this only works, of course, if you're quick with like hitting navigation, going to the right problems, like being able to go from one section to the next or one question to the next, if you're able to do that quickly. But in the video, if you saw like when I would look at a question and then it required me to go into the passage and I'm kind of looking for something in the passage and I'm still like reading it, so that information is still going in my brain, but it's not enough to answer that question, so I still flag it and I still move on.
Yeah.
But
so like
a
couple of things I noticed when I did it.
And one more thing.
Altius may be different, but the numbers you did for the discretes, I don't know if it changes per A and C test, but the discrete numbers for Altius weren't the same. It may just be that.
It could be like off by like one or two questions, and you could just go forward or backwards.
Yeah.
To check that out. But also,
because normally like it's like every 3 passages or so there's discretes.
Yeah. Also, like write down a table of like the time you should have left for like a certain passage. So that way, like, you know, when you see the time, like, you know, the most simplest way of doing that is just like you get like an hour 35 for each section, right?
Yeah.
So like when it says like you have an hour left, try to see like, you know, what passage you're on and like, you know, kind of see where you're at, if you need to like pick up the pace or if you're doing good. And so that's just one way of doing it at the halfway mark, but we can get some more accurate numbers for like the quarter mark, you know, things like that. So that's the way for you to constantly be able to keep track of what of your pace.
Yeah, I didn't really have to look at the time too much, and I think it started to run out a little bit. I started having to pick up the pace towards the end, but it was like the last 2 passages, which I thought were kind of more. The last one wasn't that hard, but there was one there that was difficult.
Yeah.
When I did it, so I'm pretty quick at like getting through, like skipping around.
Yeah.
And I found the discretes like a couple seconds. I knew they were like around 15, 20, somewhere in there. And I got to those. I did all the discretes first. I actually liked that. That helped cards a little bit because most of them were easier. And there were a few discretes that like I could not answer right away that were kind of longish. So I just flagged the yes and moved on. I tried to eliminate 2 of them if I could, and then I would just like pick whatever and then move on. I don't know if you have a strategy for that. Like if you honestly just don't know the answer and you're between 2, if you like always stick to, I don't know, like some people.
Yeah, just flag.
Just pick.
I mean, pick anything, right, and flag it, and then if you come back to it, So like if you're able to like eliminate 2 choices, you have a 50-50 chance of getting it right. So I would still flag it, but if you have other flagged questions where you're not able to eliminate it, then the one that you were able to eliminate 2 of the choices, like you would just like make that a low priority. Because the number one thing is that just that every single question is worth the same amount of points, despite there being questions that vary in terms of difficulty. Same goes for the passages. Every single passage is worth the same amount of points, but they're not the same level of difficulty. So the reason that I like to do it where you flag liberally and then like have 10 minutes or longer at the end to review is that you will have seen every single passage. If you just did it in the way that where, you know, halfway mark, you should be halfway through, that implies that, or that's under the assumption that every single passage will take the same amount of time, which isn't really the case because certain passages take a lot longer.
So that actually happened with me on FL2 because
that FL2 score,
mathy,
by the way,
there's some simple physics. Yeah,
so,
there's some like simple physics towards the end, but they took a lot of time for me, and then I couldn't get it.
Yeah,
done.
The FL2 stuff, like we hadn't worked on timing or test taking strategy when you took that, so that's why I did that like afterwards. But now that now you're going to start practicing with that type of strategy, and it's going to drastically improve the way that your performance on this, and also like you're not because I've seen this like I've taken the MCAT like a couple of times. I told you that I avoided it that one time.
I avoided my first.
yeah.
Hard.
yeah. So generally speaking, I've always seen this on the actual test where the first passage is harder than the ones that come after, and that's because they want you to be like freaked out. Like they want you to have a lot of anxiety. It's pretty nefarious of them to do that to people, but that's what they do because they know that pre-meds are very thorough, hardworking people who are perfectionists and that want to get every question right. And that is why on like here, like passage 1 doesn't look that bad, but on an actual test, like an actual one that you can't get a practice version of, I've always seen the first passage being like this tremendous hump that you have to get over, and it really changes like your entire mood for the rest of that section and for the rest of the exam.
Before I avoided, when I took it the first time, I avoided because I had such a terrible experience on the chemical process section because the first passage had some very weird mathy stuff, and it gave you like a made up unknown thing I've never seen, like formula wise, and I got really worked up about it. It was like you solved it sort of with part of the numbers. I've seen some that kind of do like that. I should have just skipped it, and then I got super bad anxiety. Some of the questions was really easy, amino acid questions. But I found lately, just talking to people and at least my experience, like the one I took, I honestly felt like was harder than anything I had encountered. I felt like it was more like Altius. Like it looked just like that. I know they kind of scale it, so I don't know how that changes test to test. Some might feel easier. The second one I took felt easier. Maybe that had a different scale, but that was frustrating. So I run out of time, so I'm definitely gonna keep working with that and jumping around.
I told you that same thing happened to me. It happened to me on the first section, CP section. It was that radioactivity question, and I spent an inordinate amount of time on it, and then I wasn't able to answer the questions for the last 2 passages, and I was like, forget it. Like I'm not even gonna I already messed up the first section, so I avoided it right away.
And they don't give you enough time to read. They really don't. Using your strategy even, I actually feel like I was reading nothing hardly, just barely enough and skimming to get enough, and it worked.
Yes.
I still got 125. I missed a few stupid things, but I got through everything and had time to go back, not really reading anything or really. I mean, some of the questions I was reading, but it was really quick and just enough to answer it.
Yeah.
They don't give you, I don't understand. They give you all this crap and you can't sit there and actually critical think long enough sometimes to get through and understand. So it's frustrating they give you these dense passages and you don't have time to really read it.
Yeah. That's why, it's good that you know now that, because look at this one right here. Look at the bottom, right? It's from, I mean, the bottom, the
Yeah, the
citation.
article.
Yeah,
Yeah.
they're research papers. So they're not really material that you're expected to really like understand like right away.
Yeah.
So you're doing the right thing in that you go to the question and you go look for like the keywords that are in that question stem, and like in the video that I showed you, like where I was doing that, I tried doing that for one of the questions and I still couldn't. It was like a question about the some peptide or something like that, or no, some protein or something. It was like basically asking about a protein and some molecule called AMC, and I was looking in the passage for it and I couldn't really find it, and because it was asking about like what, oh, it was like the molecule of AMC, and it's like at which point does it connect or whatever. And I was looking at the passage for a bit and I couldn't really find it, so then I ended up flagging it. But the important thing about that is that I was still reading parts of the passage. I was still understanding it, but I just like I would come back to that later. So anyway, so yeah, you have some better like
Yeah,
taking
I still have some
strategy.
work to do. I'm still frustrated. Sometimes I'm missing. I reviewed this already, but we can go through some of them.
Sure.
There's some things I did that frustrate me that I knew the answer for, but I don't know if it was just a little anxiety or hesitancy to go with my gut, even though I wasn't 100% sure about it. And they sometimes word biology concepts that you know a little bit off, and they kind of trick me. There was one on like this one I got right away, which is super easy. I was like, okay, well, I knew that iron from this was being oxidized, so I was like right there between two, and then I just remember they were coupled, so I got this like quick. But there were some questions. I'll just kind of try to skip around to find some. Sorry.
Yeah, and then what do you think also about? Because did you read the thing that I wrote in the OneNote?
I haven't gone back to that yet.
Oh yeah, because I go over like the strategy.
I think I gotta re-download it or log. I think I just gotta log into my account again.
Oh, okay, because yeah, I go over the strategy, like a test taking strategy in that, and it basically starts off like in basic. So here, can I actually share my screen for a sec?
Yeah.
I want to show it to you. All right, do you see this?
Okay, cool.
Yeah, so this is in your OneNote. This is Dylan MCAT, so test taking strategy, right? So here, check this out. So this is like, I wrote timing and stuff. This is like, test day, right?
Mm-hmm.
And so test day certification 4 minutes, tutorial 10 minutes. So that's 14 minutes in the beginning, right? So in that 14 minutes, this is what like I did. Like in the scrap paper that they give you, I draw out the math table. So I draw this stuff out. Like radical 2 is this, radical 3 is this. Oh yeah, in the video you kind of saw me.
Yeah, I saw you go through that.
Write it down. Yeah, and then like I didn't. Oh yeah, and then like the optics chart. So basically, oh yeah, and then also like formulas, constants, etc. And then breathe. So this is like a good thing for if you're having anxiety, because it basically activates your parasympathetic nervous system, which is the opposite of fight or flight. Like you inhale and you hold it for 4 seconds, and then you exhale for 4 seconds. So use this whenever you feel your anxiety reaching a fever pitch. Breathe, always breathe, basically. Here I wrote, I should have made the background color. Wait, what? Here, one sec. Let me try to make this more legible. Where's the background color? Font color. Okay, whatever. I wrote down, it's kind of hard to see, MCAT always makes the first passage super hard to psych out test takers. So my strategy involves going to the navigation page and doing all the discretes. First, there's 16 of them, that's 27%. So when you're done with the discretes, which should take you like no time at all, you are already done with more than a quarter of the test. So then you could go to the first passage with the confidence knowing that you're more than 3/4 done. And with the passages to go question by question, and only look at the passage if you need to. If anything takes longer than like 30 seconds, then guess and flag it. Goal is to finish with 10 minutes left or longer, so then you can spend those 10 minutes to review. Not every question is the same difficulty, but they're all the same number of points, right?
Okay.
So yeah. So yeah, the CP section, you know exactly what to do. You get a 10-minute break after for cars. So basically, once that CP section is done, you have 10 minutes of break time. Spend this break time doing breathing exercises again to center yourself. Don't think about how you did on the CP section. Don't think about questions for the CP section. It's already passed. There's nothing you can do to change how you did. So focus on the now and present. Center yourself, get ready, and then start cars. Cars is different from the rest of the sections in that you have to read the entire passage. So there's no skipping around here. I say try to be on passage 5 with 58 minutes left on the clock. Guess randomly, blah, blah, blah. 10 minutes left for review. Then you get a 30-minute break. For this break, you can do the same kind of breathing exercises. You can write stuff down on your scrap paper that'll help you, instead of whatever you have memorized, write it down on your scrap paper when you get there. So it's you don't have to memorize it anymore, and you can use that for reference. And pretty much, that kind of stuff. So okay, I'm gonna let you share your screen again.
Yeah, I'll try to show you what issues I had as we kind of go through some of this.
Yes.
Sure. Yeah, it's tough. I'm being straight with you. I have pretty bad ADHD sometimes.
Yeah.
My doctor I see my doctor and he thinks he has it as well, and he's like, yeah, when I said MCAT, I just had to go through it really fast and get through it because my mind I like to channel in and focus on one thing. I'm like, I know what this is.
Yeah.
Where is the passage? Mm-hmm. Why is all this other stuff here too?
Yeah.
I used to read the passage first because everybody tells you to read the passage first and then the question.
Yeah, they're wrong.
For me, that doesn't work.
Yeah, maybe that's the opposite of what you should do.
And they even have the AMC even put out, it was terrible. They put out this cars diagnostic, which some of the passages was really helpful, but they had this PhD go through the first few passages, and she would read it super slow and go explain the stuff. I'm just this is not helpful at all.
Yeah.
I was telling you, I don't know how some of these kids that are junior or seniors can pass this test and do well.
Yeah.
Maybe it's because they don't work and have a life or anything, but I don't understand how people do that great on this test.
Well, if you're working, it definitely makes it a lot harder.
Yeah.
Because, when I took it, I wasn't working full time or anything, because that's going to really make it tough, because you're spending most of your time at work. And if you do it before work or after work, you're going to be tired.
Yeah, definitely a lot of.
It's definitely going to be a lot harder.
For a super high score. That's why I was shooting for 505, 510, which I know is good enough for what I need to get into.
Yeah.
That's why I was wanting to work on strategy too. But yeah, there's a few things I need to review. But I'll try to skip back and try to
Yeah. So that question right there, it's talking about something that's a secondary structure.
Which one? The one before it was a back.
Yeah. So this is how I would do that. It was talking about some beta thing. This one right here, right? So this is how I would do that. So they're saying beta amyloid monomers. So I'm gonna look for that in the passage. I already found it. Do you see it?
Yeah.
There. Where? Show me.
It was up in here.
Yeah. Show me where exactly. There, right?
Yeah.
So what is it, right? It says beta sheet and polyproline helix secondary structures right there.
Okay, but here's what happened on this question. I'll tell you, because I knew it wasn't primary. I knew it was a quaternary. The reason I changed it, I saw that it said secondary, but if you read on further, it said what's responsible for reduced solubility of the beta amyloid at high concentrations. If you go on to read farther, it says, however, at sufficiently high concentrations, they undergo a dramatic conformational change to form beta sheet tertiary structure. But I know beta sheets has to do with the secondary, but that was really misleading that it said tertiary.
No, it is. You're right. It says that there's a lot of beta sheets, which is a secondary thing, within that tertiary structure. But that is that is really, I see what.
Well, I had to 50-50. I knew that's what it was, but they said at high concentration. So I was like, okay, well, at high concentrations, it is that structure. So I was confused by that. I think that was a long shot.
No, I can see why. Because they're basically saying that it forms a tertiary structure that is rich in beta sheets, and beta sheets are always secondary. So I guess they're saying, let's see.
Yep. Yeah. So you're saying the secondary before the tertiary is why it was.
Yeah, I could see how that could be tricky in terms of the wording.
That was just dumb, and I had to like move on. That kind of pissed me off though, because like, if I have something misleading like that, I'm gonna. Yeah.
So let me ask you this: so like, secondary structures, what are they? Formed by? Like, what is a cause?
I just think of H bonds
Yep.
and the beta plated sheets. There's the other thing too.
What are the H bonds between?
Alpha helices and the beta plated sheets?
Oh no, alpha helix is one type of secondary structure, and beta sheets is another type.
Yeah.
But what are they? What is the secondary structure like? How does that form? Like why does it form those shapes?
I mean, the only thing I just think of stabilization of the structure. I guess all of the bonds that are in the structure.
Okay, so basically, all right. So secondary structure, right? It's caused by hydrogen bonding between like the amine group or the like an OH group and the carbonyl of another one.
Okay.
All right. You know what? I have some notes here for this. I'm gonna see if I can copy and paste it.
Okay.
One sec. Okay, copy. Then I go to your thing. Okay. So you can't access your OneNote.
I can. I just would have to log into my mail and get in it.
Okay, got it. Because yeah, I added this stuff right here. I'm gonna share my screen again for a sec, just so you can see it. Sorry. You see it?
Yep.
All right, so it's under problems over here. So here, check this out. So, because this is very important to know. So like, primary structure is just the amino acid backbone, which is formed by dipeptide bonds, and also there's some disulfide bonds as well, basically like kind of between like cysteines and shit. Oops. Okay. And stuff.
You're good.
It determines how a protein will fold, right? Secondary structure is a spatial arrangement of amino acids near one another. So here's an alpha helix, right? Right? That's a side view. This is a top view. So you could see all these R side groups are just on the outside of it. They don't really play a role. And you see how it's a hydrogen bond, right? Between an amine and a carbonyl oxygen. Right?
Yeah. Isn't that like the same stabilization they use for like DNA molecules between the... I know it's like nitrogen oxygen groups, isn't it?
Oh, like hydrogen bonds? Yeah, between the bases. Yeah. That's the strongest type of intramolecular bond. And it's going to be FON. Think of that. FON. Those, and then like a hydrogen. So, okay. So here is an example of a anti-parallel beta-pleated sheet. So basically, same thing, like hydrogen bonding between like the H of an amine and the oxygen of the carbonyl group. All right.
Yep.
Tertiary structure is a 3D arrangement, three-dimensional. It's just one subunit, and it's mostly held together very loosely by hydrophobic and hydrophilic interactions. So this is kind of like an example. I kind of drew like this like peptide over here. So you can kind of see like some interactions here between like the negative charged aspartate and the positive charged arginine. I just drew like this kind of like green kind of like squiggly whatever line to indicate that type of salt bridge. This is a disulfide bond between two cysteines. This is whatever between two phenylalanines. This is between two serines. All right. Now, a quaternary structure only exists if there is more than one subunit. So an example of that I can think of is like hemoglobin, because that has like 2 alpha subunits and 2 beta ones. So various subunits associated together via either hydrophobic, ionic, hydrogen bonding, polar, etc. So the R chains, the interactions between the side groups, the R groups, start to become a thing in the tertiary structure. But secondary structure, it's just within the backbone. And so this is kind of like what it looks like, okay?
Yep.
All right.
All right, so I just want to
Yeah.
So this is where it started to get more difficult because this was kind of like a weird virology passage.
Yeah.
And
so let's do the strategies of. So let me look at the question. For use in humans, the mouse-human hybrid DNA construct is advantageous because. So I'm gonna look for mouse-human. So let's see.
It gets down here somewhere.
Right there we go. So basically, mouse-human 1, they hoped that it would be more effective than ZBOV, which I guess is just for mice. Because I see above, mice that had been injected with ZBOV antigens. So ZBOV is just going to be, I guess, well, I'm not sure. Let's see.
Yeah, I had to go back up.
So it has, okay.
ZBOV is a strain of the...
Oh, it's from Zaire. All right.
Yeah.
Okay, but they're basically in rodents first, right? So they're basically saying, okay, they think that it'll be more effective than ZBOV in humans. Most likely because ZBOV, I guess, they only use that in mice. That's just a guess, but who knows? Let's see. The construct contained the following components: a heavy chain mouse variable region, which cDNA, which is VH. Light chain mouse variable region cDNA, which is VL. Heavy chain human IgG1 constant region DNA, G1CH, okay? And human kappa light chain constant region DNA, CK. Alright, and then whatever, I guess you see it in figure 1 or something, but basically the constant regions are going to be from the human, I suppose, right?
Yeah, I had a really hard time with this. So then I know what they're asking at. It's kind of like a research question of why you would use that construct. I'm not super familiar with the hybrid DNA construct. I don't know if I was supposed to be.
It just means that there's some DNA from the mouse, right? Like the variable regions are from, or the variable region cDNA is from the mice.
Okay.
Do you see that? In that, yeah. Heavy chain mouse variable region cDNA, so that's DNA from the mouse.
Yeah.
Light chain variable region cDNA is also from the mouse. And then the constant region DNA is from a human.
Okay.
Yeah. So, alright, let's check out the choices. So, they're saying variable region of the mouse antibodies will bind human antigens. No. Antigens from humans will be bound. Wait, sorry. If the DNA is from mice, it'll bind mouse antigens. And the humans, whatever regions, the constant regions that are with human DNA, that'll bind to human antigens. So A won't be correct, because that is saying that mouse antibodies will bind human antigens. That can't be possible, right? So, basically, in immunology, right? An antibody is that Y-shaped thing.
Yep.
And the variable regions, which are at the ends, they are super variable, because those things need to be specifically made for whatever specific antigen that they attach to.
Okay.
So that's why, if you get, coronavirus, whatever, God forbid, you're going to have to make you're going to have to make antibodies that are specific for whatever antigens you get from coronavirus.
Yep. S protein.
What's that?
S protein.
What's S protein?
That's what's on the surface of the coronavirus. That's what the
you said asper. Wait.
S protein.
Oh,
They call
spike.
it S protein. Spike protein. Yeah.
Yeah.
Yeah. That's what the. Yeah.
Yep.
That's what this vaccine recognizes. Yeah.
Mm-hmm. Yeah.
Anyway.
Yeah.
Yeah. I just didn't understand. I actually understand immunology kind of well. It was something that interested me. I never took immunology as a class.
Yeah.
I never really touched on it. This question I just did not understand. I guess I cannot. I still don't get this. I understand what they're saying. They're like saying, why was it advantageous to use the hybrid? And I just couldn't relate the concepts to each other that well.
Okay, so basically,
I think I knew this wasn't true, but
yeah. So the mouse. All right. So if you look at, I'm going to look at the human protein turnover machinery. We'll recognize the human antibody. I don't even know what that is really trying to say.
I didn't know what that was either. That's why I picked.
Yeah. But and D is saying human antibody region will bind ZBOB, but ZBOB is the mouse antigen. So we just want to have the mouse part of the antibody bind the mouse antigens, and the human part of the antibody do human stuff. So it seems like
I think I went with this answer because I was reading it. I ignore that. But I was thinking recognize. I think I was kind of ignoring where it said human trial. I think I saw the recognized antibody region as foreign.
that would be like an autoimmune kind of reaction.
Oh, true. Wait. No, wait. Never mind. Yeah.
So, but see how B says that the mouse antibody region will bind ZBOV, which is a mouse antigen?
And basically, I see these two are variations. B and D are variations of each other, which usually is going to be one of those two. Not always.
Yeah, but the main thing to see here is just that, because ZBOV is in mice, right? Like, you see how in the paragraph above the one that we went over, it says that mice were injected with ZBOV antigens. Right? Do you see that?
Okay. Yeah.
And then these antigens were sequenced and cDNA was made from the mouse antibody light chain and heavy chain, right? So basically, like the mouse antibody region will bind ZBOV antigens, and the human antibody region will be recognized by human phagocytes. So, I was able to answer that by looking at that one paragraph, really. This one over here.
Is this one or the top?
The top just gives you some more information that says ZBOV is like for mice, basically. So overall, it like what you highlighted right there or selected right there is basically telling you that they basically made, so they said mouse hybrid, like monoclonal antibody over there, MAB, and it has the variable regions, which are coded by mice cDNA, and then the constant regions are coded by human DNA. So we just have to have the mouse part do mouse things. Since ZBOV is in mice, the mouse antibody region is going to bind to the ZBOV antigens. And then the human part is going to be recognized by human phagocytes.
Oh, I see that now. That's why I was confused with. Yeah, no, I see that now. So
for this, I just looked for that word in the passage, and that's all I looked at. So all this other crap, I did not look at.
Yeah, I didn't see it that fast. So
yeah,
we hope it'd be more effective against ZBOV in humans. Construct containing the following components: heavy chain, mouse variable. I think I was just getting really worked up with this question because I was running out. This was question 39, so I was starting to put. All right, so heavy chain, mouse variable region, light chain variable region cDNA, heavy chain.
Yeah,
I'm still
chain mouse. So just basically what you're looking at here are like I don't care about heavy chain or light chain, right? I care about mouse variable region. So the variable regions are both from mouse cDNA.
Okay, I see that.
And then there's also heavy chain and light chain for the other parts, but that's what I don't care about. But the constant regions are from human DNA. So are you allowed to highlight now?
No, I can't.
Okay. But like what I would highlight is variable region is just mouse and variable, and then human and constant. So like that's just basically like taking all this extra information. Like I don't care about heavy, I don't care about whatever cDNA, I don't care about any of that. All I see is that variable stuff region is from the mouse. Constant region is from humans. I don't care about IgG1. I don't care about kappa light chain, right? I don't care about that stuff. I'm just differentiating between the mouse stuff and the human stuff.
I mean, the only way I'm looking at it now, where I understand, is the mouse antibody region will bind ZBOB, and it says here human antibody will bind ZBOB.
Yeah, and the ZBOB stuff is from the mouse, and like you can see that in the paragraph above that. You see how it says mice that had been injected with the ZBOB antigens. Do you see that?
We're at
first sentence.
here.
Yeah.
Okay.
See that in mice that had been injected with ZBOB antigens. You see that?
Yeah.
Yeah. I didn't look at any of that stuff.
I really don't get this question.
Wait, really? Well,
Like, I just... Sorry, my dog's really screaming.
it's okay. I have like people playing loud music.
I understand why this is a bad answer choice now. I think I just picked it because I didn't understand the question at all. I mean, I understand how we generate an immune response and how vaccinations and things work. I don't know why I don't understand the construct of hybrid, like DNA, cDNA stuff. I never understand. And I'm trying to understand, like, I'm not trying to overthink the question.
Yeah.
I understand why they're saying they use the construct for whatever reason to make it more effective in humans, I guess, because...
So what is the part... So, okay. Here. Yeah.
I mean, isn't the variable region, there's like the constant region, there's the variable region, right? There's light, heavy chains.
Yeah.
I'm not trying to overthink that, but like, isn't the variable region... Is that at the top where like the
Yeah, here, let me
recognizes the
Here, let me share my screen.
Okay.
All right, so I put it here, and okay, so since it's OneNote, there's gonna be a little delay here, but I'm gonna like draw some stuff here. So basically, let's see. Okay, so this is kind of like a drawing of whatever, like an antibody, a bad drawing.
Yeah, I know what it looks like. Yeah, I get it. So yeah, I knew that's where the variable is where the antigen binds.
Yeah.
Like I know that, and I can't answer the question because like it's overwhelming.
Okay, so check this out. So all right, I see what. So like this is what you would highlight. So I wrote down. I circled mice, ZBOV antigens, right?
Okay.
And then mouse variable region, whatever. Human constant region, right? So the variable region are from the mouse, and you know that's where the antigen binds to. I mean here too,
Gotcha.
right? And the constant regions are human, right?
Mm-hmm.
So the antigen will have to bind to a part that's from mouse DNA.
Oh. Okay.
Yeah, so because the variable regions are coded by the mouse DNA.
All right, I got it. Hold on. So make sure I'm following. So in the research, they took the mice. They made sure they generated an immune response to the ZBOB antigens on the variable region, correct? So the mice are recognized. They gave this to the mice to make them recognize to generate an immune response, whatever, to the variable region, right?
Yeah.
For ZBOB. All right. So what are we doing with humans with the constant region?
So the constant regions are made by humans, and generally speaking, like, you know, basically, here. All right, so basically, this is what happens for an antibody, right? Also known as immunoglobulin, right? Like this guy. So basically, these are the variable regions. Antigens get attached to that part of the antibody, and then since the constant region is made out of a human DNA, what happens after the antigens basically the antibodies basically attach to the antigens.
Yep.
And then when they're attached, it basically signals for a macrophage to come in and phagocytose that entire thing to kill it all. I think it's called opsonization.
Yeah, I think that's right.
Yeah, here, check this out. So... Well, yeah, you kind of see this in these diagrams, right? Like, so like, yeah, so here. Why can't I find like a better picture of this over here? Here, I'll open up one of the, so like I have like these really good textbooks. So I'll just look into like one of these. So I'll do I'll search for opsonization. Okay. So here. So check this out. This is basically how it works. So you basically have. So basically, all right. So here's the antibody, right? And this is it. Basically attaches to the antigens of like a bacteria or something like that, and then once that happens, it lets a macrophage come in and eat the entire thing. And the macrophage, so just like the variable region, right, is specific for the antigen, the constant region is specific for the macrophage. So this is a human. Whoops. This is a human macrophage. So this is a mouse variable region, and it attaches to the mouse antigens. And once that's done, it's ready to be opsonized, aka the macrophage, which is a human macrophage, is going to eat the human. It's going to know what to eat because of the human constant region, and it's going to eat it. So that's how the human macrophage and the human immune system eats this entire thing. So for this question, right, I just looked at, it says mouse-human hybrid DNA. So I just looked for that, and I see that maybe like here, right here, mouse-human hybrid, right? And then I see that mouse part is the variable region, so that's the part that attaches to the antigen. So it'll have to be a mouse antigen. And then the human part is the constant region, so that's the thing that makes the human macrophage come in and phagocytose the entire thing. If you look at these choices, right, this one is saying that the variable region of the mouse antibodies will bind human antigens. That can't be true, right? If it's a variable region of a mouse, it'll have to bind mouse antigens. So this one's out. This one would be something like a autoimmune reaction, and that doesn't have anything to do with, the mouse-human hybrid. And then this is like the opposite of this. This one is saying the human antibody region will bind ZBOV antigens. Now, the human part is just the constant region, and the constant region doesn't bind to antigens. Only the variable region does. So this one is out. And then the mouse antibody region will be recognized by human phagocytes. That can't be true. But
I understand it a little better now. I think I was really overthinking this question. I honestly
yeah.
But I didn't understand the idea of a hybrid DNA construct mouse human. I was confused with this because are they basically? When I think about this, I think of a human antibody variable region and human. Are they saying they combine that?
Yeah, so they made
the mouse variable region with a human construct.
Yeah.
That's just, I guess, in my mind was implausible, but
yeah.
I'm thinking of how a human responds to any sort of foreign invader antibody cell mediated response, and I wasn't. That was throwing. I think I was getting flustered with trying to understand the experiment, and it was preventing me from answering it because now I see it. Recognize that's the way they said human phagocyte. That's. They should. I knew optimization was, and they didn't say that. That threw me off, and that makes sense now. That it was those two. So I'm still confused. If you scroll back up to.
Yeah.
All right. So, I guess I was thinking human, now just going through it, human variable region is what what creates the cell mediated response, but they're saying for some reason that I guess that's not effective, so that's why they use the mouse variable region, and somehow the constant region in humans is able to phagocytosis and
just like a variable region binds to an antigen, the constant region is what is recognized by the macrophage. So think about it like this,
I guess I was confused by. No, I just did not get this experiment. So this is me. I would have to flag it and not to guess. But so all right. So they use ZBOV to bind to the variable region on the mouse.
So here, check this out. So
Okay.
variable region, this is true all the time, okay? Variable region is the thing that binds the antigen, and the constant region is the thing that is recognized by the phagocytes.
I got you there.
Yeah.
I did not know that about the constant region because I rarely have seen questions about it. I did not know that. So
Oh yeah.
that would have been helpful.
Yeah. So if the variable region is a mouse one, it's gonna bind to a mouse antigen, and since the constant region is a human constant region, it'll get recognized by human phagocytes.
This is why this is blowing my mind because I'm thinking of the human antigen you want to recognize what's invading. But I guess this is a construct where they're saying the human one didn't work great. So they combined it with the mouse one. For some reason, it works better to bind to the variable region on the mouse. I don't understand how the constant region, though, recognizes that invader. If that makes sense.
Oh, the constant region doesn't recognize the invader.
It just phagocytes whatever it binds to. So now, whenever the antigen binds to the variable region, the constant region is going to destroy that through phagocytosis, right?
Yes.
So it didn't matter. I kept trying to separate human and the mouse as separate variable regions. Like I was thinking like we did it in this and now we're like putting it in humans. Like I wasn't thinking of it as like this was just a construct.
Oh yeah. So it's like over here, right?
Like yeah.
So and you see how like you can answer this without like I didn't read any of this or this. I didn't even look at this.
Yeah, I didn't read that either. Yeah. I went down there.
So like
but I still did.
Yeah. So because that would be overwhelming, but if you can break things down to their most like simplest elements, then you'll be able to answer everything.
Yeah, that was more simple than what I was thinking. I just did not understand the experience. So all it was mouse. So yeah, ZBOB binds. It said it in the passage. Binds to the variable.
Mice, ZBOB antigen. I made that connection. Mouse, variable region. Made that connection. Human, constant region. Made that connection. That's all I looked at. I don't know what they did in the experiment. I don't care. But I just know that for, like, this basically means mouse is going to be, the mouse part is, or the ZBOB antigen is a mouse version. Variable region is a mouse version, so they're going to bind to each other. Human part is a constant region, and that's going to bind to the human phagocytes.
Yeah, I refuse to get out of my head that the human variable region needed to recognize the invader, and the reason they did this was because that sucked basically in the mouse. Yeah, that question was really hard for me, but I understand it better now.
Yeah,
that was just really
so like rough. try answering it exactly the way that we just did now. Like for use in humans, mouse human hybrid DNA. So I look for hybrid mouse human hybrid DNA or hybrid anything mouse human hybrid. So I went from here to there, saw this, went up here, saw this, and then I think about it like this, and made these connections, and then it must be this.
Yeah, I think the only thing I was missing was that about the constant region. So
oh yeah,
like 90% of the time, all the questions, even taking micro and stuff, it was always about variable. Most people don't ask about that. So yeah, I just didn't know that. All right, that was a lot. That question really had me stumped.
Yeah. So hopefully that made sense, right?
Yeah.
Variable regions, like they're like some more information about them is that they have a lot of single nucleotide polymorphisms, SNPs, to make, because they have to be variable because like a new one has to be made for each new antigen, right?
Exactly. Yeah.
Yeah. And so and that's the thing that connects to the antigen. Whereas a constant region, it doesn't need to be different. In fact, it's worse if it's different. It needs to be the same so your human phagocytes recognize it.
Gotcha.
Yeah.
Some of these questions weren't like bad. I got on pretty quick. See, like I didn't have a problem with like this question.
Let's see. So this one I would do final mouse human MAB construct figure 2. So I'd go right to figure 2.
I did. I got it right just knowing, I
Mhm.
think, about promoters because I knew this was BS about the prokaryotes, just knowing about like the that pathway.
Yeah.
Mouse genes is initiated by a different promoter than the ones for human genes. I knew that. Oh, I knew that wasn't true because they're both. I was just thinking like eukaryotes, like mammals or whatever, because I know there's that whole different one with bacteria. That's how I knew this one was also wrong.
Yeah. Yeah, because so this is
I just knew
you can
this.
answer this
Yeah,
even without looking at Figure 1 because it tells you because we know that we know that the variable region is going to be using a promoter that is for the mice, right? And for the heavy chain so let's see. Light chain genes is initiated by a different promoter than the one used for heavy chain genes. Okay, so this actually is a little different. Let's see.
I didn't use figure 2 because I didn't understand it and I didn't want to take the time to understand it.
Yeah, it looks
was
like a mess.
So I didn't really. I couldn't follow it. Just could not.
Okay, you know
So
what? I kind of see it here though. On the right part, up top.
Here.
Oh no, on the figure, figure 2.
Oh, you know, on this part.
Yeah. So
Wait. Okay. Sorry. Yeah.
yeah. So like on the final DNA construct, like move your cursor there and then go up.
So here, then up.
Yeah, and then to the left. So okay, see, it says variable. So the VL is the variable light chain, and the CK is the kappa light chain.
Okay.
So this is like the light chain stuff, and that they use like the same promoter.
Here.
Yeah, and then VH is the mouse heavy chain, and G1CH. Oh no, I'm going to the right of that.
Here.
Oh no, the right. I'm just looking at the final DNA construct.
Oh, here.
Yeah. So now the VH is the heavy mouse whatever version, and G1CH is the heavy human constant region. So now it's not a matter of whether it came from the mice or not. It's just separating light chains and heavy chains. So the light chains all have that one promoter, and the heavy chains have the same promoter as well.
Yeah, I don't even. I think I was just I was
So
stressing
when it comes,
but I knew these were both wrong right away. I didn't even look at that. I might have skimmed it, but I was like, I don't know how to make.
So
that.
for instance, for that figure. So like, let's take a look at that figure 2. So it's a lot of crap, right? But like, you're going to learn like which thing to look at, like what's important. I didn't look at any of the things on the left. I just looked at the final DNA construct, and then I looked at this, segment over here, and I see that the light chains are in one, and the heavy chains are in the other part, and so the light chains just have the same promoter, right?
Yeah.
And the heavy chains have the same promoter, right? So there's one promoter for the light chains and one promoter that's different for the heavy chains.
Yeah.
So, like, with enough time and, like, practice and stuff, like, and if you do it the same exact way that, like, when I read it, I looked at this, you're going to be able to, like, look at something that looks complicated like that and quickly see where the answer is. Because the answer is just in that tiny region that I showed you. Like, show me again where that is, like, with your cursor.
Where? With the constant region, like down here?
Oh, no. In figure 2, what part did I use?
We used this.
Yes, that was the only part. I didn't use anything else.
Well, I had a hard time because I had to go back and look to read. I didn't know what this stood for. Like, I had to go back and see the
yeah.
Yeah, and then light chain. But I started ruling out right away because I read this and I was thinking about like Lac-Operon. And I was pretty familiar with that concept. So like, I just started reading through the answers. Like mouse genes, it's initiated by a different promoter than one from human genes. I just knew that wouldn't be true.
I would have thought that would be possibly true actually, because the previous question had us differentiate between mouse stuff and human stuff.
I just know every biology textbook I've gone through, like not to overthink content, but like they usually use Lac-Operon as an example of prokaryotic transcription. So I was thinking mammals would be very similar, so I didn't think the promoter would necessarily be different. And I did like answer the question, so that's why I ruled that out immediately.
Mammals similar to one another, you mean?
Yeah, for humans, like I just know like when they go through like I knew what this topic was talking about because they're talking about the, like there's lac operon. They use that as example like most of the time for when they talk about promoters.
Yeah.
So because the other reason is too, this one right here, all MAB genes.
Yeah, that should be
initiated by the same prokaryotic.
Yeah, so that can be it because it's prokaryotic,
but I knew it's
right?
eukaryotic. That's why I got rid of that.
Yeah.
So I knew that
good.
wasn't true. And then this didn't sound correct. So I just did not feel like that was good. So that left me with these two. Light chain is initiated by a different promoter, the one for heavy chain. The reason I went with that is I did briefly read right here where it talks about.
So I'm just going to say this, that there's no way that we could know this answer without looking at what they show us in Figure 2.
Yeah.
Because A is very plausible. It's very, very plausible. But the only reason that we know that it's not A and that it's C is just from that figure. Without the figure, we would not know that. So I want you to, like if you got something right, know exactly why you got it right. Like why the answer is right, okay?
Yeah. I just don't know in like time I would have gotten that quick enough. Like I knew to look. I knew what this was from. Like looking at that, I just had a hard time quickly like.
Oh yeah. So that's that's something that we're working on. That's why I asked you like show me where that is and show me where that is. Like I want you to I practice looking for an exact phrase or word that's in the question stem in the passage. When you can do that quickly, you're going to be at such an advantage. Because you're not going to be.
I knew to go down here right away, and I knew I needed to use this figure. Yeah. I just was not. I think at this point I was on question 42, and I probably had 20 minutes left.
Yeah. You'll get better with practice because that's pretty much the only way. And most MCAT resources, they don't talk about this, but that's what I'm going to be doing with you. I'm going to ask you where is it? Because I want you to have your eyes trained to find exactly where to look, so you don't have to process an entire passage or spend time doing that, because that's just going to be too much extra information. I believe in a low information diet, so just what you need to know, keep it nice and clean. Okay, so is this a question right here? Okay.
Yeah, I got this one right, and I did guess on this one, so maybe we should go through it.
Yeah. Okay, so basically, all right, we have. Okay, so these antibodies, right, are 150 kilodaltons, composed of 4 peptide chains, right? So when it's unbroken, it's gonna be one band, right? But it's SDS page and denaturing, so it's going to break apart. We have to know exactly how it breaks apart, though. If it's tell me what STS page would do.
That's just the one. Isn't that the one that doesn't retain its it doesn't retain its original protein structure? It breaks it down, because that was the other one. Was it non-reducing or? So I knew it was gonna break. I know it's gonna break.
So SDS page just kind of makes it have the same charge, I believe. Yeah, wait. Hold on. I have this probably in this. This is also in my Anki notebook, Anki deck, where I talk about. Yeah. Okay, I'm just gonna I'm pretty sure it just makes it have the same. Yeah, okay. It basically makes it have the same charge, I believe. Yeah. So it basically makes it uniform. It makes them all have the same charge. That's what SDS is. And then there's denaturing conditions, and then there's reducing.
Yeah.
So reducing is what breaks it up like completely. Denaturing will break it apart a little bit. Here, let me try to find my like Anki card about this. Oh wait, all right, damn, I was just there. Okay, I'll just like do a browse. Let's see. Okay, here we go. Wait, not that. Alright, so I'm gonna copy and paste this in your notebook as well. Okay, so I wrote it in your notebook. So basically, what the different types, right? So native is normal. Reducing, okay, that is what. So okay, before I talk about reducing, I'll just talk about denaturing. So denaturing basically breaks when you add the sodium dodecyl, whatever, the SDS part. So SDS page basically disrupt secondary and tertiary sulfur structures by adding a negative charge to the protein, okay? It doesn't break disulfide bonds, though. Now, denaturing like basically messes up or cancels out the structural effects, so you don't have to worry about like that really doing much. So basically, it's SDS denaturing conditions. So basically, what's happening is that there's going to be a negative charge, and secondary and tertiary structures will be like canceled out. But reducing, though, that is what breaks apart disulfide bonds.
Okay.
So remember, like between like two cysteines.
Yep.
Do you know like when people get for their hair they get a perm? Or they chemically straighten it. So when they do that, they're breaking the disulfide bridges between bonds between like the hair.
Okay.
Okay. So that is what breaks apart the disulfide bonds, which breaks the whatever tertiary structure. So we know that basically there's going to be a disruption of like secondary and tertiary, but it's not gonna. It's the disulfide bonds will still be there, right? So basically, if it was denaturing, if it was reducing, you would have 4 bands. So okay, let's say you forget everything, right? Let's just see like how this would make sense. So it's 150 kilodaltons total, right?
Yeah.
So if it broke apart into different pieces, they'll have to add up to 150.
Yeah, I knew 4 with the number 8 was dumb. Yeah.
Yeah, so A says 4 bands at 150. That would mean that it's like, what, 60 kilodaltons, 600 kilodaltons? Like, that's too much, right? So A is gone. And then, let's see. So Then 4 bands at I'm looking at D. That's 4.
It's still high.
Yeah, that's too high.
3 and 4. You actually didn't even need to know this thing because the math doesn't add up.
Exactly.
Well,
So
that's why I went 2, but I still didn't understand it that well. That's why I went 2.
Yeah, no. So okay, it's
good to
good
figure out the band part. Yeah.
Yeah. So it's good to always have multiple ways to answer a question.
Mm-hmm.
Like so basically the things that add up, so if there's 2 bands at 25 and 50, then that means that there's 2 at the 25, which adds up to 50, and then 2 at the 50, which adds up to 100.
So I was a little confused why they mentioned the 4 peptide chains, and I was confused like how denaturing conditions affected that, like how many bands you would see visually. Like I wasn't sure how to arrive at that part.
Yeah. So okay. There's a.
Maybe I was overthinking it. They just said 4 peptide chains.
Yeah.
But that's a single molecule, like IgG, like the antibodies. It's not. I guess I'm thinking of like separate proteins or something. So this is just like
you're right.
So one antibody. And
these are essentially like 2 or 4 subunits basically.
But they're all together. Like this is all one molecule, like hemoglobins for subunits.
Yeah. So it's basically that thing that I drew, like the whatever Y-shaped thing. So there's the variable regions, right? That's like one of them. There's like the there's like heavy chain variable region, and then like heavy chain of something else, and then light chain of something else. Kind of like, in the mouse thing, there's like 4 different types, right?
Yeah.
So like variable heavy and light, and constant heavy and light. Now the thing that connects the constant regions together are disulfide bonds. And that's the thing that you need reducing conditions to break.
Okay, so just to make sure I understand all of these. So for SDS page, denaturing conditions, is that always gonna reveal 2 bands? If it's like, if we're just saying like in the context of this, it would be 2 bands because you have like this one thing and it's broken up from the denaturing conditions and that would give you 2 bands. I mean, mathematically, the others didn't make sense. I was just thinking of like the bands, like how you would know.
Oh yeah, so
that versus reducing conditions, because reducing starts with like the S bridge, disulfide bridge.
Yeah. So I sent you a link of a picture of an immunoglobulin, and you can kind of get a better idea there. Because you basically see like the different kinds of stuff. Tell me when you have it open.
That you put in messages.
Yeah, in the chat.
I got it. All right. Yeah.
Okay, so basically you can see how there's overall you can see like the heavy chain, you can see the light chain, and you can see that there's disulfide bonds that connects the 2 ends.
Yeah. Yep.
So basically, just remember that reducing conditions means that the disulfide bonds break. If those broke, right, then you would have 4 bands. You would have 1 for essentially each of these.
So hold on, I am trying to overthink this. So is this example in the question non-reducing? Because I thought they say there's reducing, non-reducing.
Yeah. It'll tell you if it's reducing. So all we know is that it's SDS, so they're gonna have the same charge, negative charge, and it's denaturing, so they're gonna break apart some of the secondary and tertiary structures, but not the disulfide bond. So if you can imagine, so you see how the disulfide bonds are between, not only between the blue colored ones or whatever color that is, but there's also disulfide bonds between that and the
So I was wondering, because it's all straight in the middle, they were both connected on these ends, where they kind of branch out, they weren't.
Yeah, and then you have disulfide bonds between the pink and also on the you see the other disulfide bonds between the 2 ends. Yeah.
Yeah, here, and here.
Yeah.
So okay.
So those things don't break. So what we basically have is so it's basically gonna break everything else except for those disulfide bonds.
Okay, just hold on. Let me clarify this. Reducing is where we break the disulfide bonds, correct?
Yes. Okay.
The example we just talked through. Sorry. SDS page under denaturing conditions. So that was not reducing, correct?
Correct.
Okay. So in the case of reducing, it's breaking. It's not breaking the disulfide bridges.
Correct.
But
Yeah.
But we're gonna have two bands. But in the case of the reducing, we're gonna break those disulfide bridges, so we would expect to have 4 bands.
Correct.
Okay, I think I understand it now. There's just a couple things about that I didn't get exactly, but yeah.
Yeah. So basically, just to remember that reducing conditions are the ones that break disulfide bridges, and if it's like anything else, like SDS and whatever denaturing, it's gonna break apart everything except disulfide bridges.
Gotcha.
So you see how it said you're gonna get 2 bands. The 2 identical light chains will both migrate to the 25 kilodalton one, and the 2 identical heavy chains will both migrate to the 50 kilodalton band.
Okay.
And so A is if it separates it into individual proteins. That's only if it's reducing. And then, yeah. So the reducing part is But I'll add some notes about that in the one note.
Also, like, I'm having a really hard time retaining all this content. Like, I'm having, like, there's things I know so well just from learning them.
Yeah.
That I either got wrong a lot or practiced really hard. Like, some of this genetic stuff, like this stuff, I'm pretty good on. I practice it really hard.
Yeah.
But, like, I'm having a hard time retaining stuff because I just cannot, just with my schedule, it's hard for me to study every day. I don't know if it would be useful to use something like I can't just do content review, like knock it through. Like I know there's some things I need to go through. Like I need to relearn.
Are you using Anki? That's usually like the best way to do it.
So the only problem I had when I first started doing Anki was I had a hard time just like I would sit there for a few hours and just do a ton of cars and I couldn't keep up with it. The other problem I had with Anki was like I might remember facts about things, but then when I would take tests, like it didn't help me conceptually.
Yeah. So that's why
for like psych-soc, it didn't help me conceptually, like with chem.
Yeah.
Some of the bio stuff it did.
Yeah, that's why I always, I always say like don't just do content review, but no, like for that previous question, I didn't have SDS page memorized. I didn't have denaturing memorized, but what I did remember is that because I linked it in my brain to when you get a perm or when you straighten your hair, you break the disulfide bridges, and that was the only thing that question didn't state. So I know that the denaturing conditions and all that stuff are going to break it, so we have just the heavy chains and light chains, and those are the 2 bands. But to break it further, it's going to need to be reduced.
Okay. Another thing I was wondering too
so
is
if you
like,
make connections like that, it'll help you remember it.
Gotcha. Another thing I was wondering too, I don't know if it'd be helpful to use like there's a website called like MCAT. I can't remember, it's MCATprep.org or something. It has just like general content outlines on things.
I've used that before.
Yeah, just because
like
I don't
review.
like there's things I forget that I want to go through, that like a few topics that I need to go through, like for physics and stuff, and I just don't want to read a whole chapter because I think it's a waste of time.
Yeah, of course.
Yeah, so maybe I'll go through that a little bit like tomorrow before I take a test Saturday, just go through things I forget. There's some things I honestly just forget a lot in biology.
It's the one with like this cat anime character, MCATreview.org.
Yeah, that's it.
Yeah. I use this too. This is good because it gives you some good content without it being too verbose.
Yeah, because I do forget some stuff. It's just hard because I can't fully study. Like, the only thing I can do is take a 3-month leave of absence at work, which I really don't want to do.
Yeah.
But and I know my content could be better in a few areas. That's why I've been working more on test strategy stuff because I feel like that's paying off more.
Yeah.
Like most of the stuff I have memorized. Like I think it isn't the nonsense one with the where it generates a stop codon.
Mm-hmm.
Yeah, so I knew what that was. I know what most of these are. Like this stuff I knew pretty well.
Yeah.
This was a tough passage too that I had issues with.
Okay, so see this? I'm gonna look for that word. So let's look for that word, okay?
Yeah, I hard time finding it.
Leaflet. Yeah, that's the thing that we're gonna. There, awesome, awesome. So that basically is responsible for the generation of asymmetric blah blah blah. Let me see what that question is saying. Exceeds exceeds the rate of micelle formation. Okay, go back to that. So let's see. For the generation of asymmetric excess of lipids in the apical leaflet of the hepatocyte membrane compared to the basolateral leaflet. Okay. Let's see. These things catalyze it. And then so they basically... Alright, they said that this exceeds the rate of micelle formation. Alright, so I guess it's not going to straight up tell us. So we're going
No, that's why I couldn't understand it. So I went back up here and read about this lipid extract theory thing.
Yeah.
Like down here, it talks about bile salt micelles interact. Like nothing really helped me. I know bile salt. I know that micelle interactions and stuff by bile salts are for, but I didn't understand what this was exactly. So I went down there and it didn't really explain it that well.
Mm-hmm.
So I figured I needed to somehow use figure 1 or something, and I just could not.
All right, so let's go down to that bottom part. So basically, what I get from that is that it's asymmetric, right? And that means that there's more lipids in the apical leaflet than the basolateral one.
Okay.
So now I'm going to think that micelles are probably going to be formed in the basolateral end. So let's see if that's true. Patches. Okay, so in that picture, the apical leaflet, there's going to be more lipids there than in the and then I see the interlipid leaflet and chain exchange there. So that means that things are going to move, I believe, from the basolateral one to the apical one. Now, let's see. So, okay, micelle to vesicle transition. So that essentially seems like it means that, let's see. The rate of formation of micelles, the rate of interleaflet exchange, by that stuff exceeds the rate of micelle formation in the interstitium. So different types of theories over here. Direction of lipid flux. Okay.
So I at that point I was like, crap,
so at this point, I don't see the answer right away. So I would guess and flag.
I think I probably did that the first time. I got mad, so I came back to it. But I think I read the answer choices first. I mean, after, because I was like, okay, I don't really know what's going on. So I saw.
That's good. That's good.
Yeah, due to higher ATP levels in the apical leaflet, I knew that wasn't true, but I went down and checked just to be sure because it talks about the P-type ATPases are known to catalyze the lipid exchange.
Yeah.
And I looked at the diagram.
Yeah.
And I didn't read anything about how ATP how ATP levels would have anything to do with the formation.
Yeah, I don't see anything like
that. I just knew, and I wasn't trying to use content necessarily, but I'd never heard about that in terms of my cell formation, so I just was like, no way. And then I was between these two, but I read this one. I was like, I wasn't sure about these two, so I read this one. I was like, because lipid transport is more energetically favorable. But that was kind of talking about ATP, and I didn't see anything about. I didn't think anything about that didn't make sense to me about the energy favorability. They never talked about anything really about energy favorability. Yeah. They talked about reaction rate a little bit at the end, but it didn't make sense. So I got between these two, and I.
Oh, you know what? C seems like a good choice from what I've read.
So I went with C, and
yeah, that seems like a good choice from what I'm seeing, but I would choose that and flag it, because it does. So from what I've read, all I can see is that there's there's more. Wait, that it's asymmetric, and there's more of it in the apical leaflet than the basolateral one. So from there, that's where it's starting to like make less sense, because I see that lipid patch theory with the micelle thing in the apical end. So that's where I was
and I saw where it said lipid ratio difference, and that's sort of kind of what they were saying there.
Exactly.
I thought that made sense between the two leaflets. So
Exactly.
that's why I didn't like that about the compensating. But I realized they didn't really say, they mentioned the basolateral membrane, like compared.
Yeah.
Oh,
so yeah.
Yeah.
So okay, I would probably choose C as well, and then flag and move on. But B also kind of talks about how there's less in the basolateral leaflet than there is in the apical leaflet. So, and then I would cross out like A and D. So if I just looked at B and C, right, I'm seeing that one is saying due to the difference in the lipid ratio, and the other one is saying to compensate for not having enough in the basolateral part.
Okay. Yeah, I kind of see that. And again, this is where I started to really crap for time. These last 2 to 3 passages, I was like, crap. So
Yeah,
I was kind of
for this one, I would answer like you did and flag it.
It was weird for this. I mean, it was Altius, but they're all kind of tough. But like, I feel like the last 2 passages were harder, and they were kind of obscure. I mean, a whole passage on micelles was a little bit much, but yeah, this one I had a hard time. This passage lost me points for sure. Like, I would have been a 126, 127 probably had I not bombed these 2 sections.
Yeah. So let's see.
Instruction theory.
So then I'd go to the lipid patch. Okay. Differences in lipid content between membrane and bile micelles are the result of, let's see, this ratio, okay, lower energy neighbor-to-neighbor interaction when they're in nanodomains called patches, okay, and away from the normal mixture of interact with these patches, extracting the entire patch all at once into a bile micelle.
I had a hard time reading this passage. Like, it was very hard.
It's pretty it's pretty weird. And even understanding that figure is kind of hard.
I guess completely on this one, I gave up and I just picked D or whatever and moved on. So I was running out of time. I was like, this is not helpful. Because I had questions. At this point, I probably had maybe 18, 19 minutes, something like that. And I was like, okay, well, there was probably some questions I missed earlier on. It just wasn't worth it. So I had to move on. I did not have a chance when I had the 10 extra minutes to review. I couldn't get back to where this was. And I knew I did. It was frustrating me because I knew I was doing worse on these 2 passages, but I did get a couple points back from fixing things earlier on, just from going quick. Yeah, this one's hard.
Yeah. So, okay, let's see. There are 2 mechanisms. So one is a single lipid extraction. I suppose it means that the micelles are formed by taking individual lipids one at a time. So that's the thing at the left. It kind of makes sense. It takes So, okay. And then lipid patch. Okay, so let me see. Differences in the content between hepatocyte membrane and bile micelles are the result of micelles from different membranes with different lipid content fusing together. Bile salts. So I'm going to look for something that says patches. Bile salts inducing changes in the lipid content. No. Formation of membrane patches. I mean, they said use the word patches in lipid patch. Composed of different lipid content than the overall hepatocyte membrane. The extraction of different individual lipids from the hepatocyte membrane by self-assembled bile salt micelles. So already, I'm taking too long, so I would again guess and flag. Because this is weird. This is
I
not that
it was
easy to
it was
make
not a
sense
good
of right
question.
away.
This passage was terrible. I didn't like this, but honestly, it wasn't even good practice. Made me mad. So, but like on this one, I got so stuck on trying to, and I was running out of time.
Yeah.
So I knew so it wasn't worth it. But like, I was really trying to understand what the lipid patch extraction theory was, and it was long, and I was like, crap. I see it has to do with this ratio of the stuff, and all my cell interaction with these patches and hepatic. So like, I probably knew instinctively these were probably dumb answers. These are longer, and they made. I was trying to like make sure I've been doing this on the MCAT. I was like understanding what the hell the question is asking. So sometimes
Yeah.
I'll just skip it and like this is a result of what.
Yeah.
And I had no idea for sure.
Yeah.
I don't really know why I would. Yeah, I just guess honestly.
Yeah. So
It doesn't have the word patches in it. But
yeah, that's a very like oversimplification. But since you know, if you're running out of time, maybe. But it seems like when I read the lipid patch theory, basically they're saying that there's different types. There's a biospecific ratio of whatever what PC I think is like phospho something, phosphocholine, and then whatever cholesterol and sphingomyelin, right? So basically, they're in this patch thing, and it's a different kind of ratio or mixture that would be in the hepatocyte membrane, and then bile salts also interact, and it extracts the patch all at once and turns it into a bile micelle. Whereas single lipid is they extract an individual lipid and they do it again and they keep doing it to make it like a growing bile micelle. So in that picture, it seems like we have like that thing for single lipid extraction on the right. You see the one that has 4 bile salts.
yeah.
Show me where that is using your cursor. So,
Which one are you saying again? The what bile salts? Sorry, I'm zoning out.
Oh, in the single lipid extraction theory, the one with just the.
I'm talking about the graph.
Oh no! In that chart, in that figure. Yeah, so.
Which has the what?
So for single lipid extraction theory, right? So that's going to be on the left side. Yeah, and then basically just the 4 bile salts.
Oh yeah.
Yeah, where's
Here?
that? Good. Yeah, so that's where it starts, right? And then see how that arrow goes down and it takes one of the bile phospholipids, that black one. Yep.
Here.
It takes that and see how it adds to that up there.
Yeah.
Like follow that arrow. Show me. Yeah. Right. So it adds one, and then it goes back and it adds another. Right. So it's sequential. It adds it one at a time, and it grows. Right.
Okay.
So it grows into that thing in the middle right there. Yeah.
Here.
Now, for lipid patch, right? It basically seems like it takes like all of this crap at once.
Yeah, I get that.
And then it forms that micelle, right? Yeah.
Yeah.
So it seems like the main thing that's different between these two is one takes it one at a time and the other one takes a couple at a time. That couple is like called a patch.
Okay.
So now they're saying what accounts, or why is there a difference in the lipid content between what's on the membrane and what is eventually going to be in the micelles? And yeah, according to that theory, it says that the biospecific ratio of all this crap has a lower interaction energy state when it's in that hepatocyte membrane than when it's together in a nanodomain. So essentially it's saying that this type of like ratio of like the bile salts and the sphingomyelin and you know all that stuff is in a different energy state when it's in the hepatocyte membrane. Show me where the membrane is of the hepatocyte.
Isn't it here?
Yeah.
Or you have hepatocyte though. Oh, there we go. Okay, yeah.
Yeah, so it's in a different type of energy state when it's there compared to when it's in that patch.
Okay. Yeah, I had a hard time like trying to. Yeah, I didn't even have time to go through this that fast. Like I was,
Yeah, no,
yeah.
it's very it's taking me a while to understand it.
They
So
picked
this is
these
one of
two,
the harder
but
ones.
it was yeah, it was rough. I don't know if it's just some of their content writers. Some of the questions are hard to understand and make sense of. Like
Yeah, it's purely because of that. It's purely because it's a hard passage and it's hard questions. So overall, what I'm getting is just that there's a different ratio of all this, crap in the hepatocyte membrane compared to what's in the patch. So if I look at the choices, I see A, micelles from different membranes with different lipid content fusing together. B, bile salt inducing changes. Doesn't really say anything like that. D, extraction of different individual lipids by self-assembled bile salt micelles. I mean, that is pretty good, except for the part that says individual lipids. Because if it's individual, then it would be single lipid. Because the single lipid is, you see, you already have it selected. You see that? It says individual lipids.
yeah.
Yeah, so that's the thing. So D can't be that because that's single lipid extraction theory. But it's basically the right answer. Just if you replace individual lipid with patches, it'll be basically it.
Okay.
But yeah, this is very tough.
Yeah, I'm getting these. Oh, I got this one wrong because I was overthinking content. Like I knew this was a steroid, and I with 4 rings, and I don't know why I didn't pick cholesterol. I was so overwhelmed. Like I was running out of time, but like I was reading this, and it was talking about the cholic acid, and I didn't know what generated cholic acid was. So I was like, maybe it's phosphatidylcholine or something. It sounds so like I went up
Oh,
here.
I see what you're saying. Yeah, no.
Yeah, I knew this was a steroid and cholesterol, and I didn't go with that. This happens. I get hung up. Like
yeah,
I was
no.
over.
I could see.
Like an obvious
I can see that though because it's cholic acid. It sounds like choline.
Instead, it was being oxidized. I was just like, I don't. Yeah, and I knew the content. It just frustrates me. Like I knew the content, and I knew the steroids, 4 rings, cholesterol, and I guess. I didn't make that connection right away, and I was trying to answer the question about the cholic acid. I think they wanted you to try to like. I knew like that was dumb, and I knew it was a spectromyelin.
Well, let's say this.
This wasn't the structure.
Let's say we did think it was some version of choline. Wouldn't there be a phosphate there, for phosphatidyl or whatever?
Yeah, there probably would, but in my mindset when I'm going through this fast,
Yeah. No, I get it.
Yeah, I was between A and C, and I didn't go with my initial gut, which was wrong. I do that sometimes, but I got so bogged down with that part, like why they even mentioned that, and yeah. Because then we went back. I'm like, okay, Dylan, use the content that's in here, and I was reading through that.
I could see that.
Yeah, but I knew that spendromyelin, that wasn't the right structure, and I knew that wasn't the right structure for ATP. Yeah. I knew that wouldn't make any sense. I should have gone with cholesterol, 4 rings.
Yeah, I could see how you can connect it to the choline. So if that happens, because look, choline starts with CHO, just like cholesterol does, but I would just look at the phosphate part of it. Because I didn't thought about that first. Yeah, I should have thought of that. Yeah, that was pretty simple. Yeah, they gave immunology passage after immunology passage. Luckily, this one was pretty easy for me. Most of them I think I got right. Like I understood the style of immunology a lot better. So like this one, I didn't even read the passage, but I did extensively use background knowledge to get to it. Like if the same mice have been infected with influenza C, one would expect mice in both vaccinated and unvaccinated groups to develop symptoms of influenza. That's just because I knew a lot about vaccine, like reading vaccine research. So I just knew I knew immediately that all these were wrong. Like I just went through them. Like, yeah. Oh, I got this one wrong I feel like a dumb reason. Again, I was between these two. Blood serum drawn from the vaccinated mice at the end of study 1 likely contained what? So I went to the end of study 1, wherever that was. So I would look for the word PR8.
Look for what word?
Oh, PR8.
Yeah, so here's why I got lost. Like,
Okay, I think I see it.
I guess I should have been thinking, all right, so yeah, hold on. Oh,
Oh,
period.
I see.
Period.
It's this version of influenza Puerto Rico.
I'm just doing this because I don't.
No, that's cheating.
um. Okay, yeah, I just was confused because I knew we were using the vaccine for influenza. I think I got confused at first because with this whole strain situation, because
Uh-huh.
okay, immunized with influenza A vaccine. Oh yeah, I read influenza A / whatever 834 virus PR8, but I didn't think that's what they use. I thought they used influenza C. I think maybe it's because I read up somewhere about influenza C and it threw me off. Maybe it was down here. No, I don't know. There's somewhere I saw influenza C and it threw me off because I didn't think that was. Maybe it's just because I was going fast, but I thought I saw
Yeah.
something about influenza. I thought they were trying to treat.
So after 30 days, right? They said that they were immunized with an influenza A vaccine, right?
Yeah, I see that now. I just was going. I was rushing through this.
Yeah, and
That's
then the PR8 thing, they gave them that whatever disease or version of that, but since they were vaccinated with influenza A only, then they should have antibodies for influenza.
very.
Wait,
They
wait,
didn't
wait,
use.
wait. Sorry, it says influenza C. Okay, where is that?
No, where does it say that at?
Oh, in that choice it says influenza C.
Yeah.
Okay, so I need to see where that is.
Yeah, I was having a hard time finding it again, but something threw me off. Which is why I went back. I don't even know where it is now. I swear I found it, and that's why I went.
Yeah, what the hell? I don't see. I don't see it.
I'm just gonna Control C today because I found it yesterday when I read. Maybe it's not in here. Maybe that was a trick. Okay, well apparently that was.
What?
I swear I was
Oh,
reading it. And maybe I'm going crazy.
okay. So
Something.
I guess, yeah. So I guess that shows it. It's that doesn't. That's not even here.
Okay, well I don't. Question 54. Oops. Okay. Yeah, I knew the most. Yeah, I knew the rest of that stuff. Oh, I got this one wrong. Second guessing myself.
Oh, because of
Two.
chromatids.
Yeah, I do. You know I got it wrong. I was pissed because it's literally said diploid 2N, so I should have said 16, right?
Well, I can see what you're doing because they're talking about chromatids, which are like the sister chromatids and stuff.
And that was what I was gonna pick. The cell does has the diploid number of 16, haploid of 8,
Yeah. So like
and diploid and haploid numbers.
so yeah, you think about what happens in interphase with like G1 and then S and then G2.
Yeah.
So in the S phase, it basically replicates the entire DNA. So you have double the DNA content. So that's going to be, you know, like that's going to help you figure it out. Oh, this looks like an ATP synthase. Okay.
Yeah, most of those pretty. The discretes weren't bad. I got this one wrong. I picked the opposite answer. I knew these were wrong.
Oh
I
yeah.
knew
So what cells secrete antibodies?
I thought it was B cells,
Yep.
wasn't it?
Yeah.
Yeah, I don't know why I went T cells. I was thinking of helper T cells help secrete the antibodies. I don't know. I was thinking this. I knew both of these were. I knew both of these were wrong. I knew that the MS was a patient's own. Well, it says patient's own T cells attack the myelin sheath carrying neurons. It explains how T cell most contributes to MS. I think the question kind of threw me off at first.
Mm-hmm. There's two types of T cells though. There's the helper ones, and then there's the natural killer. The killer T cells. Right?
Yeah. I knew it wasn't a non-specific attack. That wouldn't make any sense. Like this isn't this is MS is like
Oh yeah, that choice is like it would suck if you had that non-specifically attack all cells.
yeah, that wouldn't be good. So I knew that wasn't it. And T cells activate macrophages to destroy. I knew T cells didn't activate macrophages. I don't believe that's correct.
Oh yeah, so that's how the helper T cells work.
Gotcha.
Yeah, there's those, and there's the natural killer cells or whatever, the killer T cells. That's CD8, I think. CD4 is the one that basically helps. So it's basically like the thing that, remember that drawing I drew like the immunoglobulin and then the macrophage comes in?
Yeah.
So the T cells help that macrophage go and opsonize and, destroy that. Destroy the whatever antibody and stuff.
Gotcha. Yeah. So yeah, I had it like backwards.
Yeah. So C, the answer would be what? Like instead of T cells, what would it be?
Wait.
Like what type of cell engulfs or uses the phagocytosis things?
Macrophages.
Yep. Yep.
Yep. All right. Yeah, I was just thinking of this backwards. Like,
Mm-hmm.
yeah. All right. Yeah, that was pretty much the issues I had on that. That was time a little bit on those two. They like stumps me badly.
Yeah. Well, we
But
still
the
have
stuff
like
were pretty good though. That
we still
helps.
have like 6 minutes.
Okay.
So
we can look for over a few more.
yeah.
Let me go back.
A lot of immunology stuff here.
Then pretty much, yeah, 4 or 5 passages of I didn't make any. I was like, this is BS.
Yeah.
And I did. I mean, nobody has that much experience with immunology. Like,
Yeah,
I
I always thought it was one of the harder topics, but I recently looked into it and I came up with a good analogy for the different types of cells, so that helped me out.
Oh, I just got this one wrong because I missed. This is a bad example, like me thinking things backwards. Like, based on the passage, the odds of cancer-related death following surgical removal of an HCC tumor would be greatest in patients with. You was supposed to go down here, and you could see that.
So first, I would look for surgery, and I see that there.
Yeah, it was in here.
Yeah, do you see it? Show me where it is.
It was up here. Hold on. No, I can't find it.
So kind of when you look for a word, don't read it, but just look. Yeah. So you remember how I said reading with your eyes and then reading sub-vocalizing. So if you're looking for a word, try not to read anything, but just look for it with your eyes. Okay.
Yeah.
So yeah. So surgery there. So that's the first thing that we do when we look at the question stem. We look for that word in the passage and we go from there. Okay.
Yeah.
So okay.
Which I found that pretty quickly right away.
Yeah.
But going.
Show me
there was
where,
another question.
Show me where metastasis is.
Is it in here? I didn't think it talked about metastasis, but
It's
I can
there.
look.
So this is I'm training your eyes.
Oh, here. Okay.
Look for it in another place too.
The same passage.
Yeah. So maybe above. The top of the. Top of the passage.
There we go.
Yeah. Look for. Show me where cirrhosis is. Yep. Okay. All right. So yeah, we're just gonna do that a little bit of that every time we meet, so you get good practice with that. So let's see. The question said, odds of cancer-related death following surgical removal of an HCC tumor. Okay. HCC is hepatocellular carcinoma, and let's see. When... Okay. Okay, so correlated. So vascular permeation is correlated with tumor explants. Okay, so I guess you remove the tumor during surgery and you put it somewhere. And what is it? NCB, needle core biopsy. Is that surgical explant? So the only thing that we're looking at is the one with P is less than 0.01, right?
Yeah.
Because the rest aren't statistically significant. So tumor grade, let's see what that is. Tumor grade. Liver volumes, okay. Tumor grade might be size. Are graded according to increasing invasion and metastasis. Okay, so it's because of that. So, okay, so, whatever. The grade 4 is the one where we're looking at. So so it seems like it's gonna have more invasion and metastasis and then did I see something about vascular stuff? Oh wait, no. Oh, sorry.
Earlier in the
Yeah,
passage, there was
that
one
also
day-
means that vascular permeation is bigger. So we're gonna look for just a surgical explant. So the black bar for tumor grade 4. So these things have the following: high invasion and metastasis, and high permeation. So A says preoperative vascular permeation. I see some stuff about vaccination. Small tumors. Okay. Then I have to look at what HPB and HPC is. So can you...
Oh, yeah, it's up here. And
Okay, so hepatitis, that's hepatitis and stuff. So they have... All right, I see. So, okay. If they have it, they have an increased risk of getting HCC. All right, and then due to cirrhosis decreased liver volume. All right, decreased liver volumes also decrease 3-year survival rate. All right, and then they say 3-year survival rate for patients with tumor vascularization is 62, and without it is 92. So we know the following. We know that lower liver volume, higher tumor vascularization, Hep B or C, and grade of the tumor, all those decrease survival rate. So that's what we're going to look for. So vaccination against Hep can't be it, so B is out. C says no history of Hep, and it says no vascular permeation, so that's out. A says permeation and history of Hep, that's good. D says the same thing, permeation and Hep, but it also has large tumors. So basically, the size is the difference between A and D.
Yeah, something triggered me to misread the question because I picked the opposite. Somehow, I think reading about the surgery,
Yeah.
I was thinking somehow I got it backwards, but because I knew it was these two. Because there was another question that was like triggering because it had to do with the vascular permeation and you had to use this graph too.
Yeah.
But I somehow got it that the survival rate was better. I don't know how to explain this. The surgery would be most successful if you didn't have these things going on versus your chance of death is higher. Somehow I can't remember what happened during the test, but I got it backwards.
Yeah, because it says 3-year overall survival rate, and the question is asking for the opposite.
No, because there was another question. Yeah, because there was another question.
Mm-hmm.
that asked about. I might have answered it earlier, so I got most of these right, and I don't know why I picked soups. I picked the opposite, and it really irritates me.
Yeah. Because I guess they mostly talk about survival rate,
Oh
and
yeah, it was because of this one. Yeah,
Mhm.
yeah,
Yeah. So I can see the logic of why you chose that then, because that would mean that basically you would have a higher survival rate, which is what they talk about. But the question is asking about odds of death, which is the opposite of that. But what I'm
yeah.
trying to find,
Somehow that was my reading.
yeah,
Like being slack there, going too quick. Like because I was thinking, oh, we want to increase survival rate. So if I want
hmm
that, oh, we have to do it here
yeah,
because of what was being said here. Because there was a question back where you had to figure out like
hmm
which tumor grade. I can't remember. It was back. It was a few.
was
Something
it
about the tumor
talking about the
grade.
size? Because that's what I'm trying to find now.
Oh, here it is. It was this one. I think.
Yeah.
This was it. This is why the volume of functional liver tissue is directly correlated with the 3-year OS rate. Suggests patients with a history of HPC, HPP, or HPC who develop HCC will have lower
hmm
rates than people without a history. So I was like reading about this, and yeah, it threw me off, and it messed me up on that question because
Yeah. I'm trying to
yeah,
figure out the size thing, and the most I see is just decreased functional liver volume. So I'm guessing if there's a larger tumor, you're gonna have lower functional liver volume. Because I'm trying to see the difference between A and D, because they're the same thing except D says large preoperative tumors. So I
I
guess
guess
if
we
there's
look
a
at
larger
more.
tumor, there's gonna be smaller functional liver.
I mean, yeah. I mean, I guess sort of by looking at this, are just like you're not supposed to have to know anything, but I know like the farther the grade, the more it's metastasized or whatever. So there would be more growth.
Mm-hmm.
Did it say that in the passage though?
Yeah. It says tumors are graded according to increased invasion and metastasis. So I understand that, but the other thing is just we have to deduce based on less less functional volume liver volume. It says that like up top, so we have to make like an inference here by saying
Oh,
that if there's smaller or lower functional liver volume, then it must mean that there's larger larger tumor there,
yeah. Okay.
because it doesn't say that like directly.
I was just using my knowledge of
Yeah.
tumor.
Yeah,
I,
you were because
they
they
make
are.
some big inferences. I noticed in some of their questions. Like there's some questions about vaccines like that I just had to like yeah, use kind of what I knew, which I
Mhm.
know the AMT questions aren't like that usually.
Yeah.
All right. Yeah, I did okay though. I got 125 and it was all immunology. So
Yeah. Yeah,
I missed a few
and
fun things,
Altius
but like,
stuff is
those
harder.
last few passages were just really too hard. Like I just
Yeah.
for time, if I had another 30 minutes, I probably would have gotten the other 5 or 6 questions right.
Mhm.
But all right, I'll keep working on that and go from there.
Yeah, and try like what we did today, where we look at the question first, and then we find like a keyword in the question, and then go directly to that in the passage. So try doing that, okay?
All right,
Because
sounds good.
that way you basically can answer the question by going to where you need to go. Show me where, yeah. So and then the thing where I said show me where this is and stuff. Try
Yeah.
doing that without reading it, but just looking for it. And yeah, it'll take some practice, but when you get as you get better in that, you're going to be able to really kill it in terms of time.
All right, sounds good.
All right.
All right,
Yeah. Anything else?
I think that's it.
All right, and check out that OneNote notebook. Okay.
All right, cool. Sounds good.
All
All
right.
right,
Take care,
bye
man.
bye.
All right.
All
Bye.
right, bye.